Hello and welcome to another session in the 2021 Online Student Development Conference. I'm Mariette Harcombe, Chair of the SAS Council, and with me is my fellow Council member Mpumi Maringa, and our very special guest lecturer for today is Inez Faul. Hello Inez.
Hi.
So Inez is a South African archaeologist who completed her undergraduate degree at the University of Pretoria in 2017 and her BSc honours degree at the University of the Witwatersrand. So she has recently submitted her master's thesis for examination at the same university. So the aim of her master's research was to improve the understanding of paleo-environmental conditions during the MIS 5CD at KRM. This is Klasies River site, through the analysis of ostracodes and Foraminifera. Okay, I'm not going to edit that out. That's going to stay in.
That's fine.
And contribute to a higher resolution paleo-environmental record. Klasies River Caves is one of the most significant sites and sequences in the world for modern human origin. So Inez, we look forward to your presentation on ostracodes and other things slightly unpronounceable.
Awesome. Thank you guys so much for having me here today. I really appreciate the opportunity. So yeah, thank you to all your sponsors as well. So let me actually start sharing my screen with you guys. Okay, so thank you guys so much for having me. I really appreciate the opportunity to be here with you today. As Mariette has mentioned, my project was at Klasies River main site, and my supervisors were Prof. Sarah Wirtz, Prof. Karine Anderson, and Dr. Karen van die Kerk. So for further context, let's just maybe take a second to figure out where Klasies River is. So it's over there in the red circle, as you can see. It's a sequence of caves located in the Eastern Cape province of South Africa on the Tsitsikamma coast. So the main site has two caves, which is Cave 1 and Cave 2, and has two associated overhangs, namely Cave 1A and Cave 1B. So as you can see over here at the bottom, Cave 1 is where the assemblage came from that I will be talking about today. Klasies River was one of the most intensely occupied sites in this region between 120,000 and 55,000 years ago. So what exactly are microfossils? Microfossils usually refer to the remains of protists, animals, or bacteria. They're quite tiny, so usually you will use a microscope to analyze them, and they are quite small in size. As I've mentioned, you will probably use a microscope. But in certain instances, there are things like the larger benthic Foraminifera that can be seen with the naked eye. So let me quickly take you through the types of microfossils. At the top on the left here, we see some diatoms, which are single-celled algae, or algae, depending on if you ask a Brit or a South African, or an American for that matter. Then just below that, we have an ostracode, which is what I will be discussing today. On the right at the top, we have a dinoflagellate, and at the bottom on the right over there, that is in fact not the coronavirus, but in fact a fossilized pollen. So which microfossils did I use at Klasies River? I used ostracodes and Foraminifera. The Foraminifera were initially identified by Susan Menzer in micromorphological thin sections, and the ostracodes were identified by Karine Anderson, and this was identified through the analysis of sediment. So what exactly are ostracodes? Ostracodes are tiny bivalved crustaceans. So they're also kind of colloquially known as seed shrimp, and you might have seen them in your fish tank. They have a carapace of calcium carbonate, low magnesium calcium carbonate to be even more specific. And just for more context, 2 valves equals 1 carapace. So if I do refer to a valve, I'm meaning one kind of half of this organism. Furthermore, they go through 8 molting stages from being a juvenile or in a larval stage up until they reach adulthood. And in archaeological contexts, we usually find either valves or carapaces, but not the preserved soft parts. So the preserved soft parts would be here at the bottom left. You can see all the legs and all the other bits. So just a little video to show you what an ostracode looks like when it's still alive. Sorry about the time. There we go. So there you can see the little legs and everything. I believe this one is having a snack. Awesome. So on the ostracode valves, you usually get a few very distinct morphological features to help you identify which taxon this ostracode is from. So on this one, it's a Hyperdoctoria aculeata, which usually has a very high arch and a pitted surface with some stout spines. Again, on the inside, you see some very important morphological features, such as the inner lamella, the muscle scars, which attaches the soft parts to the external carapace, and you can also see things like the hinge morphology. Next is what are Foraminifera or foraminifers. So they're also single-celled protists. They usually have either a calcareous or agglutinated test, and they can be either benthic, which means bottom-dwelling, or planktic, which means that they occur within the water column. The calcareous Foraminifera usually secrete their tests from calcite that is either low or high magnesium, and all forms of planktic Foraminifera that we are aware of today are also calcareous. So at the bottom on the left there, the one that kind of looks like a piece of popcorn that has popped or like a cartoon brain or something, that would be your planktic Foraminifera. Next, we have the agglutinated forams. These guys don't secrete their tests from calcium carbonate, but rather create their tests by sticking together a bunch of like detrital material. So this would be kind of the surrounding sediment and stuff. The agglutinated foraminiferas can survive in environments with no carbonate, which is a bit more rare for the ones that are made of calcium carbonate. So why do we use these microfossils in archaeological contexts? Well, first off, your ostracodes are extremely resilient little creatures. You can find them in nearly any type of water body. They have been so extensive that they've been found in fen soils. So fen soils are kind of just very moist soils that have come from groundwater. So that's why the soil is obviously moist. Next up, you can find them in caves, and also they have a very extensive sequence of fossils, so dating back quite a while. Next up, we can also do some geochemical analysis on these guys, which includes a stable isotope and stable carbon analysis, which I did for my project, and I will discuss in just a second. And then on the bottom right, which is a very interesting example, you can find some of these microfossils in some old pottery, and you can kind of distinguish which source the raw material came from, from which the pottery was made. So how do we extract microfossils from sediments? Well, first step would probably be selecting your sample. You want to make sure that you have a sample that's kind of big enough that you know that you will get enough organisms to be able to analyze. Next up, you're going to weigh, wash, and prepare your sample. Here on the left-hand side, we see a nest sieve, and we wash it through a 1 millimeter, 150 micron, and 63 micron sieve. So usually what this does is your 1 millimeter will take out all the chunky, due to the lack of a better word currently, chunky material like your pieces of lithics or your larger bones. And then the one that you're going to want to be looking at, or the fraction that you want to be looking at, is going to be the 150 micron. This is where you will probably find the most identifiable juveniles. So in my case, for this project, we initially were only looking for Foraminifera and not for ostracodes. So the 150 micron is kind of more ideal for the planktic foraminiferal analysis, But if you're looking at ostracodes or so, I think 125 to 250 microns could also work. Then next up, after you've washed your sediment, you're going to have to dry it at a very low temperature for a few hours. And also, sorry, before you do the washing and anything, please remember to weigh your samples every step of the way. What we've also done here at the bottom right is to kind of just loosen up the bigger classed sediments, just so that it's easier to wash and easier to work with. Next up, we have splitting and dry sieving. So the splitting works because it helps to minimize the amount of time that you're kind of spending on the sample, and it helps you to systematically work through your sample. So a conventional splitter is usually very expensive. So in my case, I was lucky enough that in 2020, an article got published about how to use a 3D printed splitter, which is what I then opted for and used in my analysis. And then next up, we just sieve it again, just to make sure that there's nothing unnecessary sticking to the sample. Next up, you're going to count and sort. So usually what I do is I try to pour out an even amount of sample on this little sorting tray that you can see on the picture here. And you go from up, down, just so that you, when you do stop counting, you kind of know where you're at. So when you're busy counting, you're also going to sort your microfossils into categories on a census count sheet. So you're going to record which species or which taxon you have, and you're going to count how much of the sample consists of which species and which split you're analyzing. Next up, as I mentioned, you're going to store your microfossils, and you're going to store them in these little micropaleontological storage cells. So very much with the exact context of your sample and what it is. So in this case, it was an ostracode called Microfossils obtusata. The methods that I used for my project included microscopy, scanning electron microscopy, a comparative database, such as the one by Martens et al. 1996 and Furstenberg et al. 2017. I used abundance data, which is relative and absolute abundances, and I also analyzed the assemblage composition. So basically, my results showed that the forams were not in situ and were likely subjected to some erosion before they entered the cave. They show signs of abnormalities due to the transport. So you can see that some of the forams don't look too good. They look a bit broken and very eroded. There's a very high number of foraminifera taxa, but low abundance of specimens. So what I mean by that is I had 10 different taxa of foraminifera that were present, but only about 2 or 3 specimens from each taxon. Furthermore, I had 4 ostracode taxa that were present, and this included Microfossils obtusata, which was my most abundant ostracode taxon. Overall, the preservation quality of the ostracode valves, excluding the ostracode that I had the least of, was adequate enough for the identification to species level. And as you can see in one of the slides that I still will show, there's an SEM image of Elyodromus sp, because I could not identify this one to genus level, that did show some dark spots that were present, and we assume that this was due to manganese staining. Okay, just for more reference, here's my abundance data, my absolute and relative abundance of ostracodes. As you can see, Microfossils obtusata and juvenile ostracodes remain the most abundant. Next up, we have the absolute and relative abundance of the Foraminifera. My most abundant taxon was Spirulina vivipara, which only shows up in BOS 2 and BOS 3. Furthermore, we analyzed the assemblage composition, and what we found was that the Klasies River assemblage closely reflected an in situ life assemblage. Obviously, whenever you do these archaeological excavations, you must keep in mind that there will be some depositional bias involved with your assemblage. But this one at Klasies does very closely resemble the life assemblage of the ostracodes. So now the next question is, how on earth did the ostracodes and foraminifera even get into the cave? So what we're assuming happened here in the case of the ostracods was probably that humans were trying to catch some fish, and the ostracods can actually survive in fish guts for a few days or so, as far as I understand. And yeah, they do this by shutting their shells very, very tightly. So what we think happened was a human probably caught the fish, and this initially deposited the assemblage of ostracods into the cave, where they then later did or were able to reproduce and live inside the cave. Also, another factor to take into consideration is storming events. So if there was a massive storm and perhaps it did overflow in the cave, then this could have deposited the ostracods into the cave. In the case of Foraminifera, because there really aren't many of them, we think that they could have been attached to shellfish that were later foraged by humans and also brought into the cave, but they could also have been brought in by wind action. And then the last theory is that in certain cases, such as the cases of an aquatic bird or waterfowl, the ostracods and or forams might even have gotten stuck to the feet of the bird. The bird might have flown into the cave, and this could maybe also have deposited the organisms into the cave. So in conclusion, the ostracod population likely survived in the cave due to the calcite formations that dripped water into the cave. So they had a fairly stable environment there to live in. Also, I should mention that my ostracods are all either fresh or brackish water ostracods. Gomphosaphira obtusata tends to be a freshwater ostracod, whereas Sarcopterodopsis aculeata, which is my second most abundant identifiable type of ostracod, can also be found in more saline conditions, but also in freshwater conditions. So the Klasies River assemblage, as I've mentioned, closely resembles a in situ life assemblage from a low energy environment. In general, environments during the deposition of Laius Moan, which dates to marine isotope stage 5C, were likely less moist than the Bos layers, which date to marine isotope stage 5D. So the marine isotope stages just refer to oscillations within glacial and interglacial cycles. Then the high abundance of ostracods that can withstand fresh and brackish waters likely supports the presence of an estuarine environment and exploitation of estuarine fish. This is also a theory that's supported by Van den Drisch. I believe it's 2004. I can make sure and just double check that it is in the references. I would please like to thank Professor Sarah Wirtz, Karin Anderson, and Karin van Niekerk, who really helped supervise this project and were amazing supervisors and were always there to support me. I would also like to thank Professor Peter Frenzel and Olga Gildiva from the University of Vienna in Germany, who helped me identify and photograph some of the ostracods. Then to Professor Jonathan Holmes from the University College London, who assisted with the stable isotope analysis of my ostracods. Professor Koen Martens and Professor David Horn for the identification of the ostracods. And one name that I did forget to add in here is Dr. Eugene Berg. I believe he's at Izika Museums, and he helped me with the identification of my benthic Foraminifera. Lastly, I would please like to thank Professor Christopher Henshelwood from the Sapiens project in Norway for his NRF bursary funding and also for assisting in the 2 lab stays that I did abroad. And also to the University of the Witwatersrand for their postgraduate merit award funding, and again, just the Sapiens project for some of their funding. Here are the references that you can please check out if you would like to read up more about the ostracods. I highly recommend Boume et al. 2003, just to get kind of a basic idea and context of the ostracods. And same with the Foraminifera, I highly recommend Murray 2006 if you really want to get a basic, good understanding of these organisms. So that concludes my lecture for the day. And thank you so much. Please send me an email if you would like to learn more or would like to ask questions. And yeah.
Cool. Thank you so much, Inez. That was a really fascinating talk. I'm just amazed that we can get so much information on paleo environments from these teeny tiny little creatures. And yeah, pretty cool work that you're doing. You have to have a very fine-tuned eye to work with these small microfossils. I'm going to open up to questions. Mpumi, do you have any questions for Inez?
Hi, yes, I do. Thank you so much. Thank you, Inez, for your presentation. I would like to know, what is the state of preservation with ostracods and Foraminifera in South Africa?
Okay, so at Klasies River, because we have an environment that's very good for preservation of things like bones and calcium carbonate, the preservation quality generally is quite good. It will depend on whether you're at an open air site or at a cave environment site, but also at the type of many factors actually, what the pH of your sediment is, what the pH of the water is it a very saline environment, is it a very acidic or whatever environment. So it genuinely does depend. I think we have a few great sites where we can get many nice forams and ostracods, most of them being cave sites though.
Okay, and then has there been any other research of ostracods or Foraminifera in South Africa in the same context that you're using?
Not in archaeological contexts, no, but I know someone in geosciences, I think it is Kate Strahan, who did an analysis of Foraminifera in salt marshes to analyze sea level changes. So she did do one, but that is a geosciences one, so not in the context of archaeology, no.
Okay, thank you.
Thank you.
Cool. From my side, how big a sample do you need to actually get the results that you got as part of your research? How many?
Okay. How many organisms?
How many organisms? Yeah.
So depending on your statistical question that you're asking, usually it is a sample size of 300 specimens to kind of get a decent idea of what your assemblage looks like. But anything more than that is also kind of preferable. So we have this kind of statistical uncertainty that the smaller your sample size, the larger your margin of error. So you kind of want to decrease that margin of error as much as you can. So generally bigger sample sizes can help, but anything less than 300 specimens, I'd say, becomes statistically a bit hazy.
Yeah, and then a slightly like an interesting question is I always ask myself, like, who first identified these tiny little creatures and decided, you know what, you little guys, I'm going to look at you and you are actually going to tell me something about ancient environments and climates. Is there a lot of research that has been done on ostracods elsewhere in the world?
Yeah, so we actually have some things dating back to the 1700s, which is a very extensive time that people have actually known about these, these microfossils. So especially in Europe, there is a fair amount of interest in this type of study. But in South Africa, I definitely think that we could be doing more to analyze these creatures in an archaeological context, especially. The more conventional way of analyzing these would be through an ocean sediment core. So the way that I did it was a bit stranger or more unconventional, I'd say. But yeah, no, I definitely think that we could use more people, especially in South Africa, like I've said, to analyze the ostracods and forams and also other types of microorganisms. So I think there are a few people doing diatoms, but yeah, we could definitely use more people.
Mpumi, any further questions from you?
Yes. I think I have like maybe like one or two. I'll see how I go. But since maybe this would be like the first time people are hearing about ostracod research, maybe not so much of forams and diatoms, but did you find any difficulties finding publications on Astrocard research?
Okay, so in a South African context, I'd say yes. So again, like I've mentioned, this is an area of study that's been quite researched in places like Europe. But in a South African context, it is a lot more difficult, especially because I think that there is one or maybe two people who analyze the freshwater ostracods. So again, here's a difference between the freshwater ostracod analysis and the marine ostracod analysis. I think there's quite a number of people who work with the marine ostracods, which are the saltwater ocean ostracods, and then the freshwater ostracods, which is different. I believe that Gun Martens is one of the people that are kind of pioneering this area of study right now. And I actually used a lot of his studies to analyze mine.
Okay. And then my last one would be, since you said that you got your sample from less conventional ways, what do you think would be the best way to give, the best advice to give excavation teams and just excavation teams? What do you think would be the best advice on how to recover these things properly, especially for research for you and any other person who would like to research ostracods, forams, and diatoms?
Okay, well, this depends on whether you know or not if there are any ostracods in your assemblage. My kind of go-to advice here would be sample as if you are kind of sampling for a geoarchaeological analysis. Take a nice bulk sample of sediment, don't sieve it. Like, just leave it be, don't sieve it, put it into little baggies, label it very, very well, weigh it, like I said, record the context and all of that. Then you can take it to the lab, start maybe washing it. Of course, another way that I think is kind of amazing is through micromorphology. So because a lot of microfossils kind of have different ways that you wash them. So the way that you'll wash to analyze diatoms is different than what you will for ostracods or forams, because diatoms are quite a bit smaller. So micromorphology does just help you kind of establish what is inside the assemblage before you go and you do all the effort or make all the effort to wash it. So yeah, I'd definitely say don't use a sieve in the field, or just save a sample for the lab and then maybe check your micromorphological thin sections.
Okay, that's good. It sounds quite similar to what I would use for the small mammals, because I find that you find more remains, micro mammal remains, when you just wet sieve rather than just sieving it, or dry sieving in the field and breaking those fragile little bones and adding more taphonomic modifications that are modern, and it just skewing everything. So I understand that, and it would be nice to just give a little heads up to people, just be like, just take a big just take a big chunk, don't sieve it, just keep it in a bag and then give it to me. So for mine, it would just be like the whole thing, because there's a lot of them and they just they just scatter everywhere. So
100%. I mean, you also worked at Klasies. I remember after washing my samples, going to you and being like, I have like 5 bottles of microfauna. Do you want to analyze them?
And I kept on saying, no, Inez, stop.
I'm over this. My work here is done. So no, the nice thing about Klasies though is that it's very rich. It's very rich in these amazing archaeological artifacts. I mean, I found so many kind of like pieces of lithic debris. Sorry to the lithics people, I'm not very familiar with the jargon, but I found so many of them, and I found teeth from, I think they're flay rats or something. So I think it's definitely worthwhile to have it and not need it, then need it and not have it, regarding to a bigger bulk sediment sample that you're taking. I mean, there will be something that you can use in some context.
Indeed. All right,
so, yeah,
I think I'm done with the questions.
Sorry, just one thing from my side. I did realize that I forgot to add the last ostracode that I said I would show in the slides. So as you can see over here, those little dots we reckon might be manganese staining, but can also be iron oxide, I've read. But yeah, just it's fairly normal. It's not uncommon. It doesn't really mean that anything's wrong with the ostracode. It's just kind of a thing, an aftermath of a depositional process.
Okay. But thank you for that. You probably would have been wondering, maybe it's a new species or something.
No, it looks like a speckled egg, actually. Like one of those little, yeah.
All right, so no more questions from me.
Your topic in terms of microfossils, and Mpumi with you with microfauna as well, reminds me of James Dietz's book In Small Things Forgotten. So this would, if you had to publish a book on the importance of looking at microfossils and microfauna, it would be in very, 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 very small things forgotten. I think that should be a title of one of your conference presentations in future.
Yeah. I actually read someone's doctorate the other day that was called All the Small Things, which is like the song from Blink-182, I believe.
Blink-182, yeah.
Yeah, and he analyzed Foraminifera from a site in Australia, and I thought that was very clever. So maybe next time I'll rename it something a bit more clever than just microfossil analysis or something.
Well, a catchy title will definitely get you some attention, especially if it's on a nice conference poster or something nice, big and visual. Okay, well, thank you very much, Inez, for spending the afternoon with us and doing this presentation for us. To the students tuning in, remember we have a short little quiz associated with this lecture. So remember, go out and do the quiz because we like to see your activity and your engagement with the material during this conference. And once again, Inez, thank you very much. And everyone must have a lovely day.
Thank you guys so much for having me again.
Bye.